last talk when we opened the entire round table. So our last speaker is David Jacob, who is going to present us the new techniques from Cordouan Technologies. And at the end of it, we're going to be then opening the entire forum for discussion with everybody. So, David, the floor is yours. You have 50 minutes.
Okay, so good afternoon, everybody. First of all, I'd like to thank CLINAM organization for giving us the opportunity to make this presentation today, which I should admit is our very first step in the medical types of conference. So for me, it's a premiere. So the title of my presentation, which is slightly different from the one described in the proceedings, is New Direct In Situ Non-Invasive Technique for monitoring biopharma product in injectable syringes. So before I start with some technical information, I'd like to say a few words about our company and myself. So I'm David Jacob. I'm the technical director of this company, which was founded about 10 years ago now. And we are located, we are located here in Bordeaux. So maybe some of you know Bordeaux by the wine, but we also produce technology, and especially optoelectronics technology. So we are located in a place that is called Route de Laser, and today we are about 12 persons, mainly highly skilled people with PhD and engineering degree. And basically our activity is create innovative and advanced solution for nanoparticle characterization, so more specifically the size and the charge of particles, and with the idea of bringing new solutions to compare to what already exists in the market. So these are examples of products that we have developed so far. So we have products for nanoparticle size analyzer, which are mostly based on dynamic light scattering, zeta potential analyzer, also based on laser Doppler electrophoresis. We have a trace detection system for nanoparticles, also with different techniques, and also some equipment for transmission electronic preparation grid. And today we have sold more than 220 units over the world. So now let me come back to the motivation of the work and this presentation. In fact, when we've discussed with some customers in the pharma industry, like Sanofi, Ipsen, or Ortiva, and asked them what was their problem, in fact, that would be related to, in particular, to particle size measurement, they mentioned the one here, which is the aggregation of proteins of micromolecules, an active principle in injectable biopharmaceutical product, which is a real problem for them because it really impairs the quality of their product and their stability over time. And this can happen at every stage of their process, even during storage. So they need to better understand and also guarantee the quality of their product. So that's why they need some tools to do that. And also, this was mentioned this morning, but the regulation also is getting more and more stringent about that. So they really need to pay attention to that. But so far, they don't have good monitoring tools. So in fact, they need new monitoring tool basically would allow them, would allow them to have a minimum sample preparation, prevent molecule destruction during preparation, eventually being non-destructive, fast and accurate, and with minimum consumables. So it was mentioned this morning that several techniques exist on the market and in the labs, and I think there was an exhaustive review by Adriel and also by Jeff. And one of them is the dynamic light scattering. So basically, just to make it quick, the principle of that. So it was mentioned it's based on monitoring the Brownian motion of particle in order to determine the diffusion coefficient of this particle, which can be related to the hydrodynamic diameter of the particles in a simple way by this equation, which was driven by, derived by Einstein and Stokes. And where basically you end up with this expression where the hydrodynamic diameter is what the size of the particle that you like to measure. It's related to this energy, thermal energy term plus this, let's say, dragging term, and then you have this D coefficient that we will measure. So in order to do that, most of DLS system, in fact, worked on the same principle. You use a laser light that is polarized, focus into a glass cuvette or plastic cuvette where you have a sample with suspension, and the light is scattered by the particles immersed into this liquid. And in fact, you collect some of this light into a detector, which can be an APD or photomultiplier. You further process the signal into a device that is a correlator, which allows then to make the mathematical treatment to infer the size distribution. So these are typical equipment that you will find today in laboratories. So this is one of our competitors, but basically you have system where you have disposable cell. You need to put a sample in the cell, put the cell in the instrument, and then you start your measurements. This is the instrument we have developed on our side, where it's an embedded cell. You put the sample in the cell by a pipette, and you close the cover and start your measurement. So basically, you will be able with this kind of instrument to measure from 1 nanometer to 5 micron, depending on the sample. DLS is a mature technique and standardized. I should admit that I don't share Hans' comments about the fate of DLS, which we had in a trash can. Just to finish on that comment, I think most of DLS is really a powerful technique when people are really educated how to use it and they know how to work and how it works and how to prepare their samples. And I think each technique has its advantages and drawbacks, so, and this has to be complementary in some way. So just to close that comment. And so you see all these equipment are benchtop equipment, so these are mostly dedicated to laboratory analysis, but they are not really fitted to what people would like to do in a process control, for example, having a non-invasive technique that would allow them to measure directly into the level. So in order to address that, there's a kind of change of paradigm, if you like, which we can summarize by this sentence, is that if you cannot bring the sample to the measurements, you have to bring the measurement to the sample. So it's with that idea in mind that we have developed this system, which is a non-invasive in situ probe. It's an optical probe. So basically, we replace this cuvette and pipetting things by this probe that you see here, where basically there's a fiber umbilical that is attached to an optical unit, and this probe contains some passive element, optical elements, which allows to focus the laser that will be used for measurements, and then collect the light scattered backward from the sample, and then make the measurements. And basically, this is an overview of the system. We have the optical unit. This is another block that contains a computer and the probe. Just quickly to show you what it looks like, to give you an idea. This is the system. So this is the optical unit, and this is the probe. So it's very small, in fact, compared to existing DLS systems. So, and size is important, by the way, when you want to deploy this kind of system in a production line, for example. So, and this is in order to implement that system for syringe measurements, what we did, in fact, we did this kind of system, so where the syringe is mounted on an holder, which can be adapted to different diameter. The probe is here, and the working distance is about 7 centimeters here. And so the light is focused into the syringe and collected backward. So as you see, there's no need to open the syringe. It's just monitored directly through the glass wall of the syringe. And this is another schematic view. So, and the scattering angle is 170 degrees. So these are the very first results that were done in collaboration with a company that is called Therapeutics, which is based in Basel. And so what they did here is they took a biopharma product from the market and tested the first thing they did, in fact, they checked the repeatability of the system. So what they did here, and this is the two graphs that you see here, they repeated 5 times the same measurements on the same sample, but removing the syringe and put it in back just to see if it was reproducible enough. And so that's what we obtained here. So you could see there's a good overlap in the size distribution, and the color correspond to the different measurements. So this is diameter expressed in nanometer, and this is intensity. And so when you convert that intensity to number, then you end up with just one peak, which is this one, which is kind of a close-up. And you realize that this big peak is a minority in terms of numbers. And they pursued some other measurements. So what they did after that, once they've checked the repeatability of the system, they did compare different biopharma coming from different origins. So, for example, here what they did is they compared the same biopharma product, but from one coming from the US and one coming from the Europe. And what they've noticed that the size distribution is quite similar, except in this region. And this was quite repeatable. So this is still not explained. That's why this interrogation mark. But it's an indication that eventually this figure here would allow to identify maybe the origin of the biopharma product. What they did after that is that they tried to see if there was a difference in the size distribution between an expired originator and a non-expired originator. And as you see, there was barely any difference. So, and the product was expired by maybe several months. And in that case, there was barely any signature. So that clearly means that maybe in that case, the size distribution might not be enough to see if the product is degrading in this specific case. Another test they did here is they compared an originator from the US with a biosimilar. And again, they find out that there was this kind of differentiation here in this region of the size distribution that we find out here. And in fact, consistently, they find that the biosimilar was very more, let's say, closer size distribution than the originator from Europe. And these are the tables that they obtained from these peaks, the 2 peaks obtained for these different measurements. They did also additional tests, but I think we have not time today but I can show you other measurements result. And they compare, for example, the in situ measurements with benchtop measurements, just to make sure that it was also consistent and that there was no kind of trade-off in the performance of the measurements by doing in situ measurements. So just to conclude my presentation, this is for us at least, it is the very first measurements we did in an open injectable biopharma. That's not the aim we had when we started to develop this product. It was more a process-oriented probe, but we realized that there was a need also in this field. These are preliminary results. We have seen that there was no significant difference between batches and ex situ measurements, even though I didn't present the result. In that case of biopharma product, there was no noticeable difference between the expired and non-expired. And also, we have noticed that eventually we would be able to determine the origin of the product by the signature of the size distribution. But this would need certainly further investigation to confirm that. For the next step of this systems, we have already started, but some measurements on flu vaccines, so they contain virus and other types of proteins and components. So the size distribution is rather more complicated. So I don't know how to interpret these peaks. I think I took 1 vaccine from the pharmacy in France, and I just measure it. So we'd like, with the help of Therapeomics, investigate this further to see some batch-to-batch variability and see if this tool can bring additional information to the people that produce this vaccine or even people that store these vaccines, we can monitor some aging effect in particular. And also, we are thinking about extending these techniques for quality control in production and online GMP process monitoring. For example, we did already some testing also on the supercritical CO2 reactor, like the one mentioned in the previous presentation. We have done several measurements also on magnetic hyperthermia experiment, where the probe allowed to measure in the presence of a magnetic field. So yeah, that's really the axis we are aiming for now for the development of that tool. So I think that's it for me. Just to finish, first I'd like to thank you all for listening to me. I'd like also to thank Professor Tudor and his team from Therapeomics for these results that I presented today.
Thank you very much, David. This is excellent.